my name is Kriesh Tivare, and my project is on researching the effect of switching magnetic fields on the growth behaviors of gram-negative Escherichia coli bacteria. So VJAS asked for a few questions, and the first question was, why did you decide on this project topic? There were many reasons why I chose this project topic specifically, but one of them was to pursue my past passion for electromagnetism. For the past 3 or so years, I've been researching related projects to electromagnetism and developing my own prototypes for world solutions. And one of my past projects was developing a system of power transmission that used LC circuits to transmit power over long distances. And this year, I started learning my biology, but more importantly, biotechnology. And I wanted to make my impact in this field. By understanding the effects of magnetic fields on bacterial growth, my goal is to research enough to develop new methods for controlling bacterial growth and potentially treating bacterial infections. The second question asked, what resources are most helpful in designing your project? So I had two main resources that helped me design my project, the first one being research articles. So reading all these scientific journals helped me build a foundation of knowledge for the topic I was getting into, as I wasn't very familiar with biotechnology at the start. And this helped me understand what kind of tools I'd be needing, what data I should expect to be receiving, and helped me get a better understanding of how I should be conducting my experiment. My second resource that was most helpful to me during designing my experiment was my past experience. As I already had past experience with electromagnetism, I already had somewhat of a foundation of electromagnetism and the topic I was getting into. And this helped me get a better understanding of how I should be manipulating the variables, what methods and materials I'll be needing for the project, and it helped me get a better understanding while designing my hypothesis. So the third question asks, in designing your project, what constraints were encountered when developing your experimental design? So one of the main constraints that I encountered was the availability of equipment. So I didn't have access to professional lab equipment, so in order to minimize this constraint, I had to innovate my own tools, one big example being my homemade incubator. The second big constraint was time. Conducting my experiments was very time consuming, and some experiments took up to 24 hours for a full run. And in order to minimize this constraint, I introduced automation in my project using my Raspberry Pi. And this automation allowed me to plan accordingly and run my projects during downtime when I wasn't able to be exactly in the lab setting. And I'll go more over my Raspberry Pi and its involvement in my project in the next question. The fourth question asks for me to comment on the strengths and limitations of my project data. So one of the main strengths was my use of a controlled environment. By controlling the environment for the temperature and magnetic field strength, I was able to minimize many of the external factors that may impact bacterial growth. The second strength I had was my automation. By introducing a Raspberry Pi into my experiment, I was able to automate processes within my experiment and save lots of time. And on top of that, I was also able to refer to the past data recorded and logged in order to see whether there were any mishaps in my project. Maybe there was a faulty temperature recorded during one of the specific hours of my project, or maybe the magnetic field strength extended its expected range. This was a very big help for me during my project. Now for the limitations. One of the main limitations during my project was limiting myself to a single bacterial cell type, which was gram-negative Escherichia coli bacteria in the K-12 strand. And there's a huge difference in gram-negative and gram-positive bacteria in their cell wall functions. And by limiting myself to one single bacterial cell type, I would need further research and data to make my conclusion for the gram-positive strains. Another limitation of my project was my experiment was limited to a field intensity range of 200 to 3000 microtesla. And this is highly dependent on the number of turns that my coil had, along with my current driver, which determined the magnetic field strength. And this can be changed and improved by experimenting with different coils, along with tweaking the current driver. Now for my recommendations. My first recommendation will be to expand on my bacterial strains. Different strains could have varying results, and to increase the generalizability of my results, I could measure and compare each bacterial strain and the impact of magnetic fields on them. Another recommendation will be to get access to a professional lab. By getting access to a professional lab, I'll be able to obtain better resources for conducting my experiment. Some of these resources could be getting better bacterial strains, getting better equipment, along with getting professional oversight, which will help me conduct my experiment more properly. The fifth question asks, what was unexpected during my project? During my project, I found that Petri dishes were getting contaminated very easily, and I underestimated the impact of environmental factors in my experiments, which made this a very unexpected finding. Contamination of Petri dishes can happen in several ways, such as from airborne bacteria or from improper handling of cultures and equipment. In some cases, the infection can't even be noticed until later stages of the experiment, in which a whole experimental run could have been wasted. The importance of maintaining an aseptic environment was an unexpected finding for me, and it highlighted the careful need for proper handling of my equipment with sterile work surfaces, sterile equipment, wearing lab gloves, and following other lab safety practices when handling my equipment. In the future, I can use my experience in order to develop better strategies for maintaining an aseptic environment and preventing contamination in the future. I will need to revise my experimental design, take additional precautions, and adjust my techniques to prevent contamination from occurring. The sixth and final question asks for me to state all of the important things I learned from the project. From this project, I learned two very important things. First was the importance of experimental design. Careful planning and execution of experimental design is critical in order to obtain reliable and reproducible results. Someone should consider all the possible variables and factors that may affect the outcome of the experiment in order to draw a valid conclusion. Second was the importance of innovating my own tools. Not everybody has access to a professional lab or professional lab-grade equipment, and I didn't let that stop me. I worked on my feet, developing my own vortex mixer, my own incubator, and introduced automation in my project using a Raspberry Pi. This helped me get a better STEM engineering mindset and will be very helpful for me in future experiments to come.